Uh, Good morning, all. Today, I'd like to share a recent work from our lab entitled Quantitative Control of Ets1 Dosage by a Multi-Enhancer Hub is Required for T-cell Function. So firstly, I'd also like to thank all the organizers for inviting us to present this talk. So with that, I would like to say that this work is recently published in the current issue of the Immunity Journal. I'd like to start with a brief introduction about how chromatin folding occurs inside the nucleus. I know that this community might not need an introduction to this, but I'll just like to state briefly that as we all know that the linear DNA is packaged to fit inside the 6 micrometer diameter of the nucleus through a series of hierarchical packaging stages. At the first, we have the nucleosomes, then we have long-range chromatin interactions called chromatin loops. We have TADs, or topologically associating domains. We have A and B compartments. And finally, we have chromosome territories. So all these chromatin structures and their functional roles have been pretty thoroughly characterized in recent literature. However, one particular structure that is not well studied and not well reported in recent literature are the hyperconnected chromatin regions. These are genomic regions that show exceptionally high level of connectivity along with other genomic regions, forming hyperconnected hubs or multi-enhancer hubs or 3D cliques. These are identified if we rank these regions according to their connectivity and the number of connections on y-axis. These regions appear above the elbow of the hockey stick plot, and these are known as the hyperconnected regions. So we wanted to ask the question, what is the role of this multiple enhancer connectivity in the context of T cells? For that, we used H3K27 acetylation high chip. H3K27 acetylation is a histone modification which marks active enhancers, and high chip measures interactions, spatial interactions between two regions. So using H3K27 acetyl high chip, we can measure enhancer-mediated long-range interactions. So using this assay in T cells, we identified a number of hyperconnected regions. Using gene ontology analysis on this region, we saw that these regions are mostly enriched in T cell receptor signaling pathway adaptive immune system, suggesting that these are regions which are actually functionally important for T cells. So we wanted to focus on one of the top most hyperconnected region, the Ets1 FLI1 locus, and we wanted to study what is the role of hyperconnectivity at this region in the context of T cells. On a closer look, we observed that this region is highly conserved between human and mice, both in terms of CTcf binding sites as well as in chromatin folding structures. Also, interestingly, there is one particular locus in this site where there are multiple sequence polymorphisms which are reported to be associated with allergic diseases in humans. So we focused on a homologous region in mice, and we named this as the Ets1 superenhancer because this region is highly connected to the nearby Ets1 gene and also has a high density of H3K27 acetyl binding. So to study how this region or the hyperconnectivity associated with this region is important, we deleted this piece of DNA in mice and we wanted to see how it affects the mice. So we wanted to see in mice how T cells are affected in terms of T cell development and differentiation. As we all know, T cell development starts from progenitors in the bone marrow, which migrates into the thymus and goes through a number of developmental stages and mature into single positive CD4 and CD8s, which are released into the periphery. In the periphery, in response to different antigenic challenges, the nice CD4s can differentiate into Th1, Th2, among other cell types. So first, in terms of T cell development, we did not observe any significant change in the number of early T cell progenitor stages between wild type and the knockout mice, suggesting that the knockout does not affect T cell development. However, in terms of T cell differentiation, when we polarized the naive CD4 cells in vitro using a combination of cytokines into either Th1, 2, or 17, we observed again there is no difference in terms of Th2 and 17. However, there was a marked change in terms of Th1 differentiation. As we can see, that the percentage of cells producing IFN gamma is significantly low in the knockout mice, indicating that there might be a Th1 defect in this mice. Next, since we observed this phenomena in vitro, we wanted to see how this affects the mice in vivo. For that, we used a colitis model in which we have a rat knockout mouse, which is devoid of T cells and B cells of its own, and is susceptible to develop colitis when it receives naive CD4 cells from a wild-type donor. So in this mice, we introduced wild-type naive CD4 cells as well as knockout naive CD4 cells and observed how it affects the mice. And interestingly, we observed that as expected, the wild-type injected mice started to lose weight, indicating development of colitis in this mouse. However, mice that were injected with knockout cells did not lose weight. They could maintain their weight at significantly higher levels than the wild-type mice, indicating some defect in them. And interestingly, we saw significantly less number of Th1 cells in this mice, suggesting that the phenomena of defective Th1 polarization that we observed in vitro was also replicated in vivo in the mice. Next, since we saw this region was associated with allergic diseases in humans, we wanted to test an allergic model in the mice. And so for that, we introduced wild type and the enhancer knockout mice to house dust mite allergens for 5 days, and we sacrificed them on day 17 to check for immune cell infiltration into the lungs. Interestingly, we observed significantly higher number of CD4, CD8s, Th2 cells, as well as eosinophils in the knockout mice compared to the wild type. These are characteristics of allergic inflammation. Thus, this indicates that the knockout mice suffers more from an allergic response due to enhanced Th2 activity, but they were unable to develop Th1 response. Thus, to summarize so far, we observe that T cell development is unaffected in our knockout mice, but Th1 differentiation is significantly affected. Naive CD4 cells from the knockout mice failed to develop colitis in the rag knockout mice. However, there is a major Th2-mediated allergic response in the knockout mice after exposure to house dust mite. Next, we wanted to see what is the exact mechanism by which the super-enhancer knockout affects this phenotype. For that, at first, we performed Hi-C on the T cells and observed that there is a significantly loss of interaction at the Ets1 FLI1 locus in the super-enhancer knockout cells compared to the wild type. This was also associated with only around 30% loss of Ets1 gene expression, whereas FLI1, the other gene in the locus, was not significantly affected. To validate this observation using a parallel approach, we used an imaging technology called oligopen 3D FISH, where we used probes against these 3 regions, the FLI1 enhancer, the Ets1 gene body, and the super enhancer region, and labeled them as E1, E2, and E3, and observed how these 3 probes behave in single cells. Here we show an example of wild type and super enhancer knockout cells where we see increased distance, or thereby indicating loss of interaction between E2 and E3 probes, which is significantly higher in the knockouts compared to wild type, thus replicating that loss of interaction that we saw in Hi-C was also reflected in terms of single cells. Next, to understand at the genome-wide level, we did a gene expression assay, and where we showed that around 33 genes were significantly downregulated in the Th1 cells after the super enhancer knockdown. And when we did a gene set enrichment analysis on these 33 genes, they were significantly enriched in Th1 pathways, thereby indicating as to why we have previously observed a Th1 defect in this mice. When we studied genome-wide chromatin folding, we also observed that there is significant loss of loops in the knockout mice in Th1 cells compared to wild type, which is exemplified here at the IFN gamma locus, where we see a strong loop forming at the IFN gamma locus in wild type cells, which disappears completely in the knockouts. Next, we wanted to verify if all these defects that we are observing is actually because of the partial loss of Ets1 gene. For that, we designed a rescue experiment where we introduced Ets1 into the superenhancer knockout cells using retroviral constructs. And on overexpressing Ets1, we saw that the cells started producing more IFN gamma with almost similar levels compared to the wild type cells, indicating that the defect that we are observing might be actually because of partial loss of Ets1 in the knockout cells. Next, to understand how the loss of Ets1 might be actually regulating the 3D genome topology of the Th1 cells, we looked into the binding pattern of the chromatin architectural protein CTcf. As we can see here, the regions that lost Ets1 binding after the superenhancer knockout, a fraction of those also correspondingly lost CTcf binding, thus indicating that Ets1 level might be actually controlling the 3D genome topology of Th1 cells in a CTcf-dependent manner. Pile-up plot analysis of Ets1 and CTcf co-bound regions also showed significant loss of local interactions around this region in the knockout cells. Thus, to summarize, we saw that in wild-type cells, in wild-type mice, due to the hyperconnected hub formation at the Ets1 FLI1 locus, there is optimum Ets1 levels. But in the superenhancer knockout, due to the loss of connectivity, there is around 30% loss of Ets1 expression, which fails to recruit CTcf to important Th1 specific chromatin loci, and loss of Th1 leading to loss of Th1 specific chromatin folding, for example, at IFN gamma locus, which makes the mice prone to allergic diseases. Based on our summary, we had a journal cover art idea for our paper, where we show that loss of loops leads to lower volume of Ets1 and making the mice fall sick. But whenever the hyperconnected region is intact, it modulates, it increases the volume of Ets1, and the mice are all happy and dancing. Moving on to some of the limitations of our work and current project that we are working on. One particular thing that I wanted to point it out is that the superenhancer region that we deleted also harbored a non-coding RNA. So we wanted to ask the question if the non-coding RNA had actually any role in the hyperconnected hub formation or the Th1 defect that we saw. So for that, we designed 2 more CRISPR knockout mice. In the first one, we deleted the promoter of the non-coding RNA while keeping the DNA of the non-coding RNA intact. In the second one, we inserted a poly-A sequence just after the transcription start of the non-coding RNA. With both the mice, we wanted to keep the DNA region intact with the idea of reducing the transcript levels of the non-coding RNA to see if the RNA actually had any role in the defects that we saw. Another limitation in our imaging experiments using oligopaint was that we were limited to probing only 3 different regions in the locus using oligopaint, but we need to map the entire chromatin region to visualize the hyperconnected hub formation at single allele resolution. To overcome this limitation, we are currently using ORCA, or optical reconstruction of chromatin architecture, which sequentially probes the entire chromatin region to visually trace the chromatin folding pattern and hub formation within individual cells. This is initial data from our lab where we show a correlation matrix of IC data generated from 0.5 million T cells and only 5,000 cells and similar T cells on ORCA data generated from it. As we can see, there is almost 72% correlation between the Hi-C plot and the ORCA data that we have. We can also visualize individual chromatin traces in single alleles as exemplified here. So this is an ongoing project in our lab, and so far we have very promising results. With that, I would like to thank all members of our lab, including Golnaz, for being an excellent mentor. I would like to thank all our collaborators and funding sources, and I would like to thank you all for listening, and I'll be happy to take any questions.